Okay, let's look at the basic attributes of cells. You have the plasma membrane that encloses the cell and allows interactions between the cell and its environment. Another name for the plasma membrane is the cell membrane. And it's a pretty thin membrane, but it does have two layers. It's known as a phospholipid bilayer, meaning it's made up of two layers of phospholipids. There's also proteins embedded in this bilayer. It includes things like carrier and channel proteins, receptor proteins, and it allows the plasma membrane to not only just be a boundary, but to participate in exchange and responses and look at the environment of the cell. Here's a quick look at the plasma membrane. You can see it's mainly made up of these yellow structures, which are the phospholipids. They are bilayered, so you have two layers with their tails facing inward and the heads facing outward. The heads like to be near a watery environment. They're hydrophilic. So they can be facing the outside and the inside of the cell. Whereas the tails are hydrophobic, so they face the inside of the bilayer. And embedded throughout are proteins like channel proteins and carriers and receptors. And they're generally attached to something on the outside or inside to provide the cell structure. All cells also contain, on the inside, cytoplasm, and that contains the fluid and the structures inside the plasma membrane, but also outside the nucleus. The fluid portion of the cytoplasm we've talked about before, that's called cytosol, and you find that in both eukaryotic and prokaryotic cells. Also inside of cells is a cytoskeleton, so a cell skeleton to provide support, transport, and also to allow cells to move and change shape. They also play a role in cell division. The cytoskeletal elements of cells consist of things like microtubules and intermediate filaments and even microfilaments. All cells also have genetic material. DNA is the hereditary instructions that RNA utilizes to guide the construction of proteins and make other cell parts. DNA stands for deoxyribonucleic acids, and segments of DNA are called genes. And genes store the instructions for making all of the parts of a cell, specifically proteins. Ribonucleic acid, or RNA, is chemically similar to DNA, and it helps process those genetic instructions. We process genetic instructions through RNA that come from DNA on ribosomes, which can be thought of as the protein workbenches of the cell. Prokaryotic cells also have a relatively simple internal structure, but they still have ribosomes. They still have cytosol. Even though they're generally less than 5 micrometers in diameter and they're unicellular, they do make up two of life's domains, archaea and bacteria. So here's a quick look at prokaryotic cells. They have a nucleoid region, not a true nucleus, but that's where they keep their genetic material. They also have other pieces of DNA, like plasmids, that may or may not encode for special properties. They have ribosomes, and all this is floating inside the cell membrane in the cytosol. But they also have more than just a plasma membrane. They have a cell wall, and sometimes they have a capsule or slime layer to protect them. So they're still complex, even though their structure is simpler than that of eukaryotic cells. So let's look closely at some of those specialized cytoplasmic structures. So first, we have a distinct nucleoid region for prokaryotic cells. It's generally a circular chromosome, but it's still DNA. They also might have those extra pieces of DNA called plasmids. You can see that in the image below, and it's in yellow. Now, prokaryotic cells also have distinctive surface features that we don't necessarily see on eukaryotic cells. They have a cell wall. We do see that on plant cells, but not animal cells in domain eukarya. But they might also have, again, a slime layer or a capsule to protect them. They're going to use these extra layers to help kind of maintain their shape. On that slime layer or on that capsule, they might have pili. Singular would be pilus. Those are surface proteins that project from the cell walls of many bacteria, and there's two different types. There's attachment pili and sex pili. Attachment pili help the bacteria or archaea adhere to surfaces, whereas the sex pili are going to form connections between two different adjacent bacteria. They're going to help them transfer genetic material, usually those smaller plasmids. And some bacteria and some archaea possess flagella. That's going to allow them to move about in their environment, and the flagella generally rotate to propel the cell through fluid environments. So again, let's come back to the image of a prokaryotic cell. We want to be able to locate the nucleoid region, plasmid, flagellum, and also the plasma membrane in the presence of a cell wall and a capsule or slime layer. You can also even see the adherence or attachment pili in this image. But let's go to eukaryotic cells next. They make up the bodies of multicellular organisms, especially in domain eukarya. They contain membrane enclosed organelles that are specialized for a particular function. This contributes to an overwhelming amount of complexity for eukaryotic cells. For example, take this plant cell. It has a cell membrane, it has a cell wall, so it has things we've seen thus far. But it also has membrane enclosed spaces besides the nucleus. The nucleus is important, it contains genetic material, it holds it in place. We actually have to have RNA go into the nucleus and then exit in order to help get the instructions to make protein. But there's also the Golgi apparatus, which is the post office for the cell, helps manufacture, modify, and package things that go and move throughout the cell, or maybe even exit the cell. There's something like the cytosol, which holds free ribosomes, or we could find attached ribosomes on the rough endoplasmic reticulum that makes proteins. This is the plant cell, so it has a chloroplast, and that's going to facilitate photosynthesis. There's a lot more to look at and to talk about for domain eukarya. Animal cells are also similar. Now, we might not have organelles like a chloroplast, but we have those like mitochondria and the Golgi and the rough endoplasmic reticulum, and of course, the nucleus. So let's talk more about these eukaryotic cells. Well, first, what's around eukaryotic cells, especially animal and plant cells? Well, of course, we have the plasma membrane, but in between cells, outside of their internal environment, there's an area called the extracellular matrix, or ECM. This is oftentimes reinforced with things like cell adhesion molecules, or adherence proteins, or proteoglycan molecules, or collagen fibers. And that actually creates a supporting kind of adhesive mesh, or maybe even you could call it a polysaccharide gel, that supports these cells in their environment. In plants, they have a cell wall, but for many animal cells and those without a cell wall, we need this extra structure provided by the extracellular matrix to keep cells shape. So here is an image of that extracellular matrix. Up at the top, where it's labeled interstitial fluid, we're looking at the outside of the cell. Some of these adhesion proteins or proteoglycans or collagen, they actually are touching or adhered to the outside of the cell, but they thicken the fluid that's outside of a cell, creating a more gel-forming substance. Now, if we go back into the cell, we have the nucleus, and the nucleus is the control center of the eukaryotic cell. It contains DNA, it stores information in segments of DNA called genes. Those provide the instructions for how to construct different cell parts and to direct countless chemical reactions, generally by creating proteins as modulators. And the nucleus has three major parts. We have the nuclear envelope, which surrounds the nucleus. Inside, we have the genetic material, DNA, kind of wound up with protein. That's called chromatin. And the nucleolus, which is a dense staining region that helps create things like ribosomal RNA. So here we can see those three parts of the nucleus. You have the nuclear envelope, which is double layered, and it has pores to allow things to enter and exit the nucleus, but the DNA never exits. You can see the chromatin. Think of chromatin as unwound DNA. It's unwound, so it's easier to read. But it is going to be a composite of DNA and protein. And then you have the nucleolus, this central kind of dark staining space. This is where ribosomal RNA is made. And the nuclear envelope actually allows, with its pores, selective exchange of materials. That nuclear envelope, while it's double layered, the pores are perforations, and they're going to regulate passage of large molecules. They're going to help ribosomal RNA get out into the cytosol and help form ribosomes. Ribosomes can actually be found throughout eukaryotic cells. They stud the outer nuclear membrane, they're found in the rough endoplasmic reticulum, some are free. And that chromatin that's inside of the nucleus is not unimportant, it's the DNA. Now, it's DNA associated with proteins, and early observers thought that the nucleus name and the nuclear material, chromatin, was kind of a misnomer. We weren't sure at first if DNA was really the genetic material. But what chromatin consists of, and what we now know is the genetic material and the dictator of protein instructions, is chromosomes, made up of DNA and associated proteins called histones. And when a cell is not dividing, the chromosomes cannot be distinguished because they're unwound. They're not succinct and compact and supercoiled. But during cell division, they do supercoil, they do condense, and they become very visible. So here are some visible chromosomes. Even the chromatin that's indicated in the image on the left is not as discernible, and it's in the early stages of condensing, which is why we can see it at all. But it is important to know about DNA and chromatin and chromosomes. And just to summarize a lot of the material, let's make sure we got our definitions down. So DNA is deoxyribonucleic acid. DNA is found in the nucleus of eukaryotic cells, and segments of that DNA are called genes. Each segment of DNA, called a gene, dictates the formation of a protein. Proteins can be structural and supportive, but they can also be functional and then carry out different chemical reactions within the cell. So these genes are specific sequences of nucleotides that provide instructions for the synthesis of proteins. And we make proteins at ribosomes. So the DNA is in the nucleus, the ribosomes are in the cytoplasm. So how do we get that DNA instruction to the ribosome? Well, to do so, you have to use a go-between molecule. And that go-between molecule is RNA. Turning DNA into RNA is called transcription, and turning RNA into protein is called a translation. These are two processes in the molecular dogma of cellular biology that talk about how to turn genetic information into a protein sequence to make a protein that has a more functional blueprint for what should happen inside the cell. And the reason that transcription is turning DNA into RNA is because they're both going to still be the same biological molecule. They're very similar. Just like if you were to transcribe notes from this recorded lecture, those notes would be similar to what I'm saying. Turning DNA into RNA is making something similar. But when you translate, you turn something from one language to another. And a nucleic acid like RNA is not really similar to a protein. They're different molecular languages. So turning an RNA molecule into a protein, that is translation. What you can see here with this process is that if we didn't have RNA, we couldn't translate the genetic code from DNA into a protein. RNA is very important. There's actually 3 different types of RNA. There's mRNA, the messenger, tRNA, the transfer, and rRNA, which is ribosomal RNA. And they all participate in different parts of transcription and translation. So protein synthesis is key in cells, but not if we can't read the DNA confined to the nucleus. Thankfully, DNA can be converted into RNA, specifically mRNA, called messenger RNA, and that messenger can leave the nucleus and go to the cytoplasm and head towards a ribosome. Ribosomes are protein-making machines, so they're a special organelle that even prokaryotic cells have. So again, this is just the basics of protein synthesis, but it's important to know how cells function, and this is a major part of their function. But we've looked at the other parts of a nucleus, the nuclear envelope, the chromatin, how to use that chromatin to make proteins, but let's not forget the nucleolus. That is the site of ribosome assembly. Ribosomes are actually made up of rRNA, or ribosomal RNA, and proteins. So we want to make them because we want to make proteins, because they're structural and supportive and functional in cells, carry out many chemical reactions. But where do we get the components of a ribosome? Well, we get it from the nucleolus. It's a dark staining region in the central kind of area of the nucleus, and it's really important for making those ribosome components. So let's review. The nucleus, found in eukaryotic cells, has 3 parts: a perforated nuclear envelope, chromatin, which is DNA plus proteins, and a nucleolus that makes the ribosomal RNA. All right, we're going to talk more about ribosomes next. And ribosomes are composed of, now we know, rRNA and proteins, ribosomal RNA and proteins. And where did that rRNA come from? The nucleolus. Now, we've been calling ribosomes the protein-making machines. They really are going to be that workbench for protein synthesis. Some appear singly or free in the cytoplasm, but most ribosomes are found on certain organelles, like the nuclear envelope or the endoplasmic reticulum, called rough endoplasmic reticulum. Any place a ribosome is, that structure probably has something to do functionally with making proteins. Some ribosomes are actually strung into polyribosomes. So here we can see a couple of areas where you can find a ribosome. You can see the endoplasmic reticulum is highlighted, and there are smooth and rough versions of endoplasmic reticulum. The rough is called rough endoplasmic reticulum because it has ribosomes and appears bumpy. Ribosomes are those double subunits made up of protein and ribosomal RNA, and they read messenger RNA to make a protein. In order to make a protein very quickly and modify it and just have an abundance of what we need in a cell for function purposes, some of the ribosomes exist as a polyribosome. We have many ribosomes together, helping to constantly translate the mRNA into a protein, so we have an abundance or a bunch of it. So that covers some of the basic parts of a eukaryotic cell, but we're not even close to done. There's many other membrane enclosed areas or organelles within eukaryotic cells. So we're going to be looking at them next, like mitochondria, and also looking at what those do, plus or compared to chloroplasts. So make sure to watch that video as well.